So good afternoon here and good morning to our colleagues in Africa. Hope that you can see me and also can hear me. So we would like to welcome you all for this important seminar. And today the seminar topic is sequencing populations to find causal genetic variants. And our guest speaker is Professor Richard Mott. He is coming from University College London from the Institute of Genetics. He is a well-done professor of computational and statistical genetics in the Research Department of Genetics, Evolution and Environment at University College of London. Some of you were present there in our CEG meeting room, so already mentioned a few points, like he was previously at Wellcome Trust Center for Human Genetics and a professor by research at Oxford University. And he has been working for several years on the different topics, including physical mapping, and for the human genome, where he developed a suite of software tools for construction and validation of physical maps way back in 1995. Then he moved to Sanger Center. There he worked on the DNA sequence assembly. His background is mathematics, very good in bioinformatics, so developed software which can automatically analyze the sequencing trace data and edit DNA sequence assemblies. And these tools were used to accelerate the sequence production. He wrote several tools, including the CAF tools assembly pipeline, and also developed the software for spliced alignment of ESTs to genomic DNA. He was with the Wellcome Trust Center from 1999 to 2015 and working on the human genetics, where he served as head of bioinformatics and statistical genetics. In 2010, he stepped down to concentrate on his own research, leading a group on working on quantitative genetics in plants and mice. So he moved to UCL in November 2015. And then he has been working with this large number of collaborators. And recently he is having this good project from the GCRF on magic populations in wheat, together with naive, rice with irri, and with chickpea with us. So we are working with him, and I also visited his lab few weeks back, and now we already had some good interaction. So he is having great expertise to develop the methods for mapping in outbred stock of mice, which is heterogeneous stock. He developed the HAPPY software package used for high resolution QTL mapping, which led to the identification of quantitative trait genes underlying behavioral variation in mice. And as I mentioned, he's having a lot of collaboration on the different aspects, including on mice, which they call it collaborative cross. And and also in Arabidopsis, they developed the magic population. So I think he got really great experience in these different areas. So we will be heavily benefiting from his experience from Richard. So please join me in welcoming Richard for this seminar. And yeah, floor is yours, Richard, please.
Thank you very much, Steve, and thank you for all the hospitality I've had since I've been at ICRASAT. I've enjoyed talking to many of you. So today I'm going to talk about what we can learn by sequencing populations, and in particular how we can use sequence data to try and find causal variants. So there are a number of reasons why we might want to sequence a population. We might simply be interested in cataloging all the genetic variation that's present. The more likely reason is that we want to perform some kind of genome-wide association study. We want to be able to find genetic variants which are associated with phenotypes. We may also be interested in finding structural variation in the population. We may also be interested in questions about the history of that population, what its structure is, how it arose. We may be interested in activities of selection and drift. So I'm only going to talk about the first two, or rather the first three of those items, but I'll also give you a sense of things that you could use. Can you hear me okay? Okay. So if you come from a human genetics background, you're going to have a different viewpoint about how you should sequence populations than maybe is present in crop genomics. But I want to talk you through how we would go about this at present in humans, and then see how this could be applied to crops. So the basic idea is that we want to sequence a large population of individuals, but that's expensive. Okay, so instead we kind of compromise. We sequence a limited number of individuals at high coverage. That would be typically maybe in the case of humans, we might be sequencing them at 30x coverage, because we have to remember humans are heterozygous, unlike many crop genomes. So we would sequence a small number, who we would designate as what we call a reference panel, okay, or rather a haplotype reference panel. And these are often, in the case of humans, they would be cell lines. Okay, so we're not talking about living human beings. These would be cell lines that you can order from a repository. So the advantage of sequencing a small number of people at high coverage is that you can create a haplotype reference panel, okay, and you will see most of this variation in a relatively small number of individuals. Okay, so this is one of the most kind of unintuitive or surprising results of population genetics theory, which says even if you have a population of millions of individuals, it's very likely that if you look at one particular place in the genome, there's only a few dozen common haplotypes there. Okay, so you can actually capture most of the haplotype variation by sequencing a small number of representatives. So, okay, you do that, and then you'd move. And then you would move on to a sample where you actually have some phenotype data, and this time you would use a genotyping chip with maybe 1 million or 2 million SNPs on that chip. And then you use the fact that the chromosomes of your sample are going to be mosaics of the haplotype reference panel to essentially fill in the gaps, to make it look as if you actually sequenced the entire population. And then you can perform a genome-wide association on what we call the imputed genotypes. So the imputation here just means predicting missing genotypes. So this is a sketch of how we would go about doing that. This is from a review paper by Jonathan Marchini. So down here we have some genotypes. So each row is one individual, and we're coding genotypes with the numbers 0, 1, and 2, which tells you how many copies of the reference allele you're carrying at that particular SNP. Okay, so the idea is that we've genotyped on an array just a few places. Okay, we know there are some other SNPs present, but we haven't genotyped them, so we don't know their values. Okay, so this individual along the top has 1 copy of the reference allele here, and 3 unknowns. Then 1 copy, 0 copies, 2 copies, 2 copies, 2 copies, 0 copies. Okay. So we want to fill in these gaps. We want to fill in the question marks. And over here we have, from a previous sequencing experiment, we have a reference set of haplotypes for this region. Okay. And here we have 3 individuals whose 2 chromosomes at this locus have been decomposed into their haplotype mosaics. Okay, so we basically found that this individual here looks like the red haplotype, and then followed by the green haplotype on 1 chromosome, and the light orange followed by the light blue over this region. So once we know this haplotype mosaic, then we can essentially fill in the gaps. We just look them up in the haplotype, and then we get a complete fully sequenced haplotype. And the reason for doing this is that if you did a GWAS just on the genotypes that were in the on the genotyping chip, you might see something like this. So this is genome position. This is significance of association with a phenotype, measured usually as a negative log 10 p-value. So the higher up values are more significant. Basically, there doesn't seem to be much going on there. When you add in all these imputed variants, which are in orangey red, then you actually get something which is associated. So that's the idea behind human genetics as it's currently performed. What happens when you move to other species, or maybe to populations which are a long way away from the reference set of haplotypes that you have? Well, you immediately hit a problem, several problems in fact. You may only have a reference genome. You may have nothing else. Okay, so you won't have a haplotype reference panel that somebody else has kindly generated for you. You may not have any genotyping arrays already made for you, and it may be in any case that your subpopulation is rather different from the reference, so that the sequence variation that you think might be segregating actually is incorrect or incomplete. So what do you do? Well, you can just sequence the entire population. So again, you can sequence a few individuals at high coverage, and then you sequence your population sample at very low coverage. And here I've got 3 human genomes. This is from a project called Converge, which I'll talk a bit about in a moment. Each individual was sequenced at just 1.5x coverage. Okay, and at first sight, that seems to be an insanely low coverage, because you're going to get huge gaps where there are no reads covering a particular individual, and it seems to be very unlikely that you would sample both alleles at any given site. So the problem we face, therefore, is that this is one small fragment of an individual's genome. The SNPs that person is carrying are labeled here AA, TT, CT. Here we have a heterozygote, another heterozygote, so on. And here are reads covering some of those SNPs. Okay, so based on just that read information, we can't reliably call these genotypes just looking at these reads. Okay, we know that one of the 2 alleles at this site is an A. We have no information about this place. Again, we know one of these alleles is an A. Moving over here, we're lucky we actually sampled both alleles from both chromosomes, so we know that that's CT. Over here, though, we only know that's an A. Over here, we have 2 reads covering the one place, but they both happen to sample the same allele C, so we don't know whether this is a heterozygote or homozygote. Okay. So that's the best we could do if we just take the sequence for that individual in isolation from everything else. What we'd like to do, of course, is to impute all these genotypes. But the great attraction of low coverage population sequencing is that it is cheap. It's certainly competitive now with genotyping arrays. You have multiple SNPs on the same read, okay, like over here, and that gives you some information about haplotypes, so that's quite useful. But the key observation is that if you sequence enough individuals and you pile up all those reads simultaneously, you've actually got a massive coverage of the genome. So then you can see immediately which sites are segregating, and with a bit of clever informatics, you can actually impute all the sequence variants. So I'm going to talk about a program called Stitch, which a PhD student Robbie Davies came up with, and I think this method has a lot of application to the types of populations that you're sequencing at the moment to add ICRISAT. So Robbie's method makes some fairly standard assumptions about the data. Okay, we assume that our population has descended from a number of unknown founders. Okay. So if this is a magic population, of course that's true by construction. There are 8 founders typically. Okay, so K will be 8. In the case of humans, K might be say 50 or 60, but of course we don't know. We have no way of knowing what those founder haplotypes are. And it's important to emphasize that this is the number of founder haplotypes. So the number of founder individuals would be half that number, or a quarter, I suppose, if you're dealing with a tetraploid. So what value of K should we pick? Well, that's going to depend on the species and on its population history. And you can think of making a magic population as essentially bottlenecking down to just 8 individuals and then expanding out again. And the key observation is each chromosome of the population is going to be a mosaic of the founders. Okay, so we go and sequence as many individuals as we can using short reads. The coverage we need is going to depend on the value of K and on number of samples we have. So essentially there's a bit of a trade-off here, and you're going to have to use a bit of knowledge about how the population arose to choose a sensible value for your coverage. But typically, I would say, less than 1x is fine for magic populations. 1 to 2x is probably fine for outbred natural populations. So the number of individuals that you want to sequence, okay, will depend also on what it is you're going to do with the data. So if you're looking at human populations, we expect any genetic effects we pick up in a GWAS to be minuscule in their effects. So we need thousands or tens of thousands of individuals. If you're dealing perhaps with crops, you may need fewer alleles, individuals, but you will be in the best position to determine that. So the problem we face is we need to be able to estimate both what we call the founder haplotype space, that's just another name for the haplotype reference panel, and the mosaics of each individual. Okay, if we know those two pieces of information, then we can impute each individual's genotype. So before I get on to that, I just want to make clear what we mean by imputation. Okay, so we mean something slightly different from just saying, oh, the person's genotype at this position is CT. What we actually do is calculate or impute a genotype dosage, okay, which is the number of reference alleles that individual is carrying at that particular SNP. So that's going to be 0, 1, or 2 for a diploid individual. The other point is that because although in theory it's 0, 1, or 2, in practice, an imputation algorithm may give you a number which is dispersed. Okay, so this is like an ideal scenario. So here we're looking at a regression of dosage on a phenotype, and this is the regression line. So this is how we would test for association between phenotype and SNP. We're basically doing a linear regression on dosage. Okay, but it may well be that our imputation algorithm doesn't give us nice, clean dosages. They may be more spread out. Okay, so just bear that in mind. Okay, so what imputation is going to do is it's going to estimate these dosages as the expected number of reference alleles carried by an individual. So this is the only equation you're going to see in this talk. So don't worry too much if you don't like mathematics. But the basic idea is the expected number of reference alleles carried by individual I at a particular SNP G is found by summing over all the possible pairs of founder haplotypes that could be present. Okay, so here we have the probability that the founders are H and K at this SNP G and this individual I, times the probability that haplotype H has a reference allele at that SNP, plus the probability that the founder haplotype K has a reference at G. So that's basically all that imputation is. Okay. As I mentioned, you won't necessarily get exactly 0, 1, or 2. This is typically what you would see for a well-imputed SNP. Okay, so you're going to get some individuals with imputed values close to 0, some close to 1, some close to 2. And we measure how well this imputation is working by essentially doing a correlation between the array dosages and genotypes that we might measure by some other method, such as using a SNP array. Okay. So the key observation to analyzing any kind of chromosomal data is that it's linear. Okay, and that means that the natural way of modeling a chromosome statistically is with what's called a hidden Markov model, which basically says your chromosome is a mosaic of regions which have different statistical properties. Okay, so what Stitch does is it models each chromosome as a hidden Markov model. And in a hidden Markov model, you have to have hidden states, and these are the haplotypes. Okay, so the haplotype mosaic is what's hidden about the data. The SNP calls in the sequence reads that align to that individual's reference genome are the observed data. Okay, so this is what we see, and this is what we want. Okay, and we also have a reference genome sequence. Okay, and we have our low coverage sequence data, which has been aligned to the reference. So the basic idea behind Stitch is an iteration. So you would first of all call all the sequence variants you can in the reads which are aligned to the reference using a standard caller like GATK. Then you initialize your ancestral haplotypes at random. You just you just toss a coin to decide what the reference allele is, or whether it's the alternate allele in a given haplotype in a given position. Once you've got a set of reference haplotypes, even if they're pretty poor, you can go ahead and run a hidden Markov model across each individual's genome to work out what the haplotype mosaic would be given that choice of reference haplotypes. Okay. That then lets you update what those reference haplotypes are, okay, using an expectation maximization algorithm, okay, and you repeat this process. So you re-estimate the haplotypes, you then re-estimate the mosaic, you re-estimate the haplotypes mosaic, and the process converges after about 40 or so cycles. Okay. So this is a diagram that Robbie generated. I had argued with Robbie that this was a totally uninformative way of showing what was happening, but he was insistent this was the way to show you. So I'll take you through it. So this is for a population of mice. I'll talk more about these in a minute. Called CFW, and these have only 4 founders, or rather, they have two founder individuals, so 4 founder haplotypes. So we start off, okay, with 4 haplotype mosaics. So each of these blocks represents 1 founder, okay, and the height of each bar represents the probability that it's going to emit a reference allele at that particular position. So the SNP positions are the different bars going on the x-axis, and the y-axis measures the probability of emitting the reference. Okay, so we basically use a random number generator to simulate some random haplotypes. Down here we have just 2 individuals. This was actually run on 2000. I'm just showing you 2 individuals. Here are the reads that were aligned to these 2 samples. I should say this population was sampled at very low coverage, only 0.1x. Given this information, okay, which looks very, very sparse, you can start to update the what the haplotype should be. So over here we've labeled the possible haplotypes with colors, okay, and they correspond to these chromosomes here. So we've made the assumption, rather the hidden Markov model's made the assumption that this individual is the orange and green haplotype. This one is homozygous for the orange haplotype. Okay. Given this mosaic, we can update this, and we keep going round and round and round until at the end our imputed haplotypes look like this. So haplotype number 1 is emitting the alternate allele almost entirely. Haplotype 4 is emitting the reference allele. Haplotypes 2 and 3 are doing something more complicated. Okay, and then this gives us our imputed haplotypes, and also our imputed dosages. So because this individual is predicted to be this and this, okay, and this individual is imputed to be this and this. Okay, so I'm going to take you through sort of three examples of using this. First, the reason we wrote this was for a population of commercially available outbred mice called CFW. So these are bred in huge numbers by commercial mouse breeding companies for use by the pharmaceutical industry. They're used for drug testing, for example. So essentially, what the pharmaceutical companies want is a reasonably uniform population of mice. These are not inbred. These are heterozygous animals, but they're kept in very clean conditions. They're cheap. Okay, that's the other thing. So it's thought that sometime in the 1960s, this stock was bottlenecked down to two individuals, but they didn't keep the DNA. Okay. So we don't know how they were founded exactly. And it's about 100 generations since then. So the population has sort of grown and then been maintained in a large, very stable size for some time. We think the effective population size of this colony is around 1,000. Okay, and they don't have much population structure. So this perhaps resembles what a population on a small island might look like. You know, if a couple of founder rodents got onto the island, and then there was no other influx, and they were just able to breed, they would eventually be limited by the food supply and levels of predation. Okay, so, and you would end up with a population looking like this. So we sequenced 2,000 of these animals at 0.15x coverage. If you pile up all the reads, you get a 350x coverage of the mouse genome. So the mouse genome is about the same size as the human genome. It's about 3 gigabases. And there are about 5.7 million SNPs segregating in this population. So that's quite a small number, okay, compared to what's segregating, say, in the human population. And nearly all of these are actually known from previous sequencing studies of mouse inbred strains. So we can actually tag these SNPs using just a subset of 360,000 SNPs. We phenotyped each mouse for over 100 traits. So the aim here is to reduce costs. So each mouse went through a standardized phenotyping protocol for a whole range of different measures, behavioral, disease models, standard blood, immune phenotypes. And we could map about 255 quantitative trait loci for 156 different, for 92 different phenotypes, which represent 156 different regions of the genome. So this is a sort of superposition of all the Manhattan plots for all of those trait associations. I'm not going to talk about the QTLs in detail, but what was interesting is we do find a number of clusters of hotspots of pleiotropy. Like we also find some regions of this genome which effectively were fixed. So chromosome 16 contains hardly any sequence variation, and consequently contains no QTLs. That's almost certainly because the founders just happen to be identical chromosome 16. Okay, so this was all done using the imputed genotypes from Stitch. So the question is, how accurate were those imputations? So we genotyped about 70 of the mice on an array called the Illumina Megamuga. We also sequenced 10 of the mice at about 10x coverage. So looking at this this diagram gives you a sense of how well you're imputing across a large number of SNPs. Looking at this diagram tells you about SNPs which are maybe are private to the population. Okay. So Stitch you can run essentially in two modes. I'm not going to talk about the differences here, they don't really matter. What matters is the fact that the correlation R squared between our imputed SNPs and the actual SNPs as determined by both the array and by high coverage sequencing, the R squareds are very high, they're very close to 1. We also ran a couple of competing methods, Beagle and FindHap, and they don't perform anything like as well. This is not really a fair comparison though, because these programs weren't designed to run on ultra low coverage sequencing. And we were essentially asked to do this by the referees. But I don't think this is a fair thing to do. Beagle is an excellent program. It's just not designed to do this. Okay, so the other thing to notice is that imputation accuracy goes down when the allele frequency of the imputed variant is small, which is not surprising. Essentially, it means there are hardly any individuals carrying that sequence variant in the population, so you have virtually no data to impute from. Okay, so you need to have, your pileup has to actually capture all the variation, and you have to have some individuals which actually did have that sequence variant. We can also play around with how many founders we think were present. So we can have 2 haplotypes, 3, 4, and so forth. This is the average squared correlation between the imputed dosages and the real dosages. And you can see here that the sweet spot is indeed about k equals 4, which is what we expect. The runtime of the algorithm starts to increase dramatically as you increase the number of haplotypes. So you want to choose k to be as small as possible without sacrificing efficiency. This thing here means we tested whether seeing 2 SNPs on the same read was giving us useful information about haplotype reconstruction. So we basically artificially broke up all the reads, so they only ever had one SNP on them. And you can see there's a reduction in accuracy when you do that. And I should say Stitch runs just off the BAM files, okay, of the aligned reads. The second example is from human genetics. So this is a study of major depression in China, the Converge project. So we collected about 12,000 samples from about 50 hospitals across China. Okay, and the problem is that we don't know much about the ancestry of humans compared to a nice, well-controlled mouse population. So we don't have a set of founders in any real sense of the word. All we have really is a sort of haplotype space. Nonetheless, we can use this approach. Okay. So we sequenced the genomes of about 5,000 Chinese women with major depression and 5,000 matched controls. These are all unrelated individuals as far as possible. And each genome was covered at about 1.7x. We imputed the sequence variants, and we actually find two loci over here which have associations with depression, and we could replicate these in a different population. So these associations are probably real. I'm not going to talk about them too much, except one of them covers a gene called SIRT1, which is quite an interesting candidate. You may have read it has an impact on lifespan in nematodes. It also has a role in mitochondrial regulation. So I want to talk a bit about how we did the imputation for the human genomes. We played around with different values for k, and we find that k equals 40 is a good compromise. Okay, and we did the imputation two ways. So the first was we used Beagle with a reference panel. Okay, so we told Beagle what haplotypes were present using 1,000 genomes, which is a human haplotype reference source. Okay, and then we ran Stitch. Okay, without a reference panel. So Stitch has to estimate everything just from the data. We also used a clever speed up, which reduces the complexity of the algorithm from being quadratic in the number of founders to being linear in the number of founders. But I'm not going to go into the details of that here. Okay, and then we also sequenced some individuals at high coverage, and we genotyped some individuals on an array, so we could measure accuracy. So if you do that, you basically get a similar picture to what we saw before, except that this time Beagle isn't doing too badly. Okay, if you tell Beagle what the reference panel is, then it does nearly as well as Stitch. Okay, this other software find hat doesn't do so well. So this is for SNP arrays, and this is for sequence genomes. And again, we see a drop off at low frequency SNPs. Okay, and we can play around with different numbers of founders. And again, we find a sweet spot sort of around here, around 30 or 40 founders. Okay. We can also play around with downsampling our data, and both in terms of coverage of individuals and in number of individuals, and then test or estimate how well we would do. So this is quite useful for designing experiments. Okay, I'm not going to talk too much about that, but if you go to the paper, there is an analysis there. The third population I want to talk about is an Arabidopsis magic population. Okay, and most of this is going to be about structural variants. Okay, so as you may well know, Arabidopsis is the primary model plant. It's got a fairly compact genome. It's 120 megabases in 5 chromosomes. It's a small plant, so you can grow them in growth chambers very easily, or outside in greenhouses. And it's highly variable genetically. So we made a magic population for Arabidopsis. This was published back in 2009. And our population was unusual in that we started with 19 founders rather than 8. Okay, this was basically to try to capture as much of the worldwide genetic variation for Arabidopsis as we could. So the breeding scheme looks something like this. This looks horribly complicated, but basically we have a diallele cross, an F1 of all possible pairings of the 19 founders. That's represented by this matrix here. And then the idea is you've got a particular magic line down here, and if you trace back, it comes from picking pairs from this array at random. So you cross those, and then you pick. So this individual is formed by picking that cell, and then you cross it with this and this to produce this. Okay, and then you continue and you start to self. After about 6 generations, you have an individual which is pretty much inbred, and which will be a mosaic of at least some of the founders. Okay, and at the time we published on this, we couldn't sequence the genomes, so we genotyped them at about 1,000 SNPs, and this is the haplotype mosaic for just one magic line. So these are the 5 chromosomes. The red lines indicate the chromosome boundaries. Yes, that's right. The 19 founders are here. The shade of grey you see is the probability that that individual is descended from that particular founder at that particular place. So this individual looks like this. It's fairly cleanly predicted. And over here we have something which looks rather messy. So this is a place where we're not sure what the founders are, but it's pretty clear it's either this one or this one, for example. And this may either be due to residual heterozygosity or because the two founder haplotypes are actually identical by state at that locus. Okay, we then went on and we sequenced these lines at very low coverage, 0.3x. If you run stitch on the data, again without the reference panel, without telling it the founders, we get a very high concordance with SNP genotypes, 98%. So that's all I'm going to say about stitch on the magic Arabidopsis. The reason for talking about Arabidopsis is because of structural variation. So this is work done by another PhD student, Martha Imprielu, and this was published earlier this year. So you may think it's hopeless trying to call structural variants in a population sequenced at 0.3x. Okay, and in some sense it is. Okay, you will never be able to predict structural variants in one individual in isolation. Okay, just on 0.3x coverage. But again, if you look across all the individuals simultaneously, there is actually a lot of information. The second thing you can do is that you can sort of reduce your ambition slightly, and it kind of pays dividends at the end. So rather than saying I want to predict a structural variant and I want to know where the breakpoints are, and I want to know is it an inversion, is it a tandem duplication, whatever, I just want to know is it likely there's a structural variant present at a particular locus in a particular individual. And the way you can do that is you start off in the way that all structural variant predicting programs start, which is that you look for reads mapping to the genome which have some kind of anomalous mapping characteristic. So if we have an inversion, then rather than having a read pair pointing inwards, they go like that. Okay. If we have a duplication, we may get a sudden increase in coverage. If we have a deletion, we may get an apparently abnormally large insert size for the read. So we divide the genome up into bins of width 10 kb. In the case of the Arabidopsis genome, that means we have essentially 12,000 segments of the genome. Okay, and we just count how many read mapping anomalies there are in each bin for each individual. And we normalize that by the average coverage for that individual. And we can measure mapping anomalies in a number of different ways. We used 6 measures. And then you can estimate how much variation there is in that read mapping signature across all individuals at each locus in the genome. And this is what you see for chromosome 1. Okay, ignore the colors for the moment. So the y-axis is showing you how much variation there is in read mapping anomaly in each bin. Okay, and you can see there are some spikes which are much higher, and these correspond to structurally variant regions. Okay, and there's essentially a large cluster of them here, which is on top of the centromere, which is what you kind of would expect. So I'm going to spend some time on this slide, so bear with me. The important thing is that you can actually deduce some very unexpected information by thinking about how structural variants arose in the past and what the pattern is you expect to see in the present population. So let's focus first of all at the top of this slide. Okay, and let's suppose there's some ancestral individual, and there are two here. I'm showing 2 chromosomal regions, and one of them is going to be called the source, the other is going to be called the sink. And imagine that in this individual, this segment was transposed over to here. Okay, so this is what it looks like after the transposition has occurred. There's now a breakpoint here, and there's an insertion here. Okay, and let's suppose that the untransposed version of this individual made it through to the present day population and became the reference genome. Okay, we don't have to make that assumption, but for the moment, let's just make that assumption. Okay, and then let's think about what our present-day population sample is going to look like. We would expect to see all 4 possible configurations. Okay, there'll be some individuals which were actually identical to the reference. Okay, so these are essentially unchanged. There are some individuals which will have the breakpoint over here and the insertion over there, i.e. corresponding to this individual. There'll be some, because of random assortment of chromosomes and recombination, where we have the breakpoint here, but we have the version of the chromosome without the insertion. Okay, and there'll be some where we have the original and the insertion. Okay. Now, if you were to sequence all these 4 possible types of individual, if we were aligning the reads to the true genomes, in each case, all our reads would align perfectly. Okay. Problem is, we align to the reference genome. We align to the wrong genome. And if we do that, then we get a characteristic set of anomalies. And in particular, you get an anomaly at one place if you're carrying a particular haplotype at a different place. So that tells you where the transposition has occurred. So it tells you where the sink was and where the source was. Okay, so we need to look for associations between the presence of a structural variant trait at one locus and a particular haplotype at a different locus. So that's the key observation. And it may well be that those 2 loci actually coincide. So that would correspond to, say, just a local inversion. Okay, but that inversion will be characteristic of occurring on a particular haplotype. But if it's a transposition, then we will see read mapping anomalies at one place only when you're carrying a particular haplotype at a different place. Okay, so that tells you that you've got a transposition. So that basically maps the problem of identifying structural variants to a genetic mapping problem. We now have to do a GWAS for every possible structural variant trait. Okay, and remember there are going to be 12,000 of them because we divided the genome into 12,000 pieces. So it's a bit like an expression QTL study. You can think of it that way. If you do that, we end up with about 6,500 what we call structural variant QTLs. And remarkably, 25% of them are transpositions. Okay, that was a much higher figure than we expected. This is just a subset of those structural variant traits. So there are 19 founders in this population, and I just took one of those founders and have mapped those structural variants which are associated with that particular founder. Okay, don't worry about the different colors. What you need to worry about really, or think about, are these arrows. So this tells you that a piece has been moved from here to here in this founder. You can see the 5 chromosomes. You can see there's a large number of structural variants within each centromere, and there's a fair amount of traffic between the chromosomes. So you can also use this to identify causal genes. So one of the problems you have with magic type populations, as I'm sure you're aware, is the mapping resolution is not single gene. Okay, in the Arabidopsis we get down to about 200 kilobases, but Arabidopsis has a gene every 5 kb. So yeah, we have a lot of genes to consider. The structural variant traits have a very different characteristic. They don't really have linkage to equilibrium in the same sense at all. So let me take you through what's going on here. Let's focus first of all on the top panel, and I'm showing you 3 GWAS, if you like. This is just for one chromosome. First of all, we have a GWAS for germination time, which is the orange curve. That has a log P, which is genome-wise significant, about 7, I think. Okay. We then asked which structural variant traits have a direct correlation with germination time. Okay, and that's what's plotted here. Okay, and you can see there are essentially three structural variant traits which have a strong direct correlation with germination. So what we're doing is we are looking across the 470 odd individuals, and we're correlating the germination time for that magic line with and the number of structural variants, the value of a structural variant trait of that individual at that particular locus. So let's focus on the trait which is highest. So if we look at the GWAS for this trait, it's by this thing here. So this is a cis structural variant trait. Okay, so it's going to turn out to be a deletion. So you can see that there is a very similar pattern, except that the green is higher than the orange. So if you regress out this structural variant trait on germination time, you get this blue curve here, which is no longer significant. Okay, so in other words, the structural variant trait here can explain germination time at this locus. If we now zoom in to that particular region, okay, and we plot all the 19 founders, okay, and this is a small region of that genome. The blue bars indicate the different 10 kb bins, so this is 10 kilobases. The intensity of the shade indicates the numbers of reads mapping, okay, to each founder, and we can see there's essentially a very nice deletion covering 3 genes. Okay, so in this case, we can say with some confidence that one of these 3 genes, or possibly all of them, is responsible for this QTL. And the nice thing is, as I said, you don't get, whereas in the GWAS you've got LD extending out a long way, the structural variant traits are much more isolated. Okay, so if you are lucky, as in this case, you get a very clear signal as to where the causal variant is. It's not always as nice as that. This is another QTL for fungal resistance. The plots work in the same way. Here we have a structural variant trait which is a little bit broader, which is highly associated with fungal resistance. That you can see that the founders are much less clear cut, but there is a difference between this group of founders and this group. Okay, and this is telling you something which I think is generally true, which is that structural variation is highly polymorphic, probably because it's associated with transposons. So it's generally not going to be the case that you have a biallelic structural variant segregating. What you've probably got is the continual turnover of structural variation at the same locus. So the different founders of your population may all carry slightly different versions of the same thing. Okay, so this approach is quite nice in that respect, because it doesn't insist, it doesn't try to call a structural variant, so it doesn't get stuck doing a problem which doesn't really matter. Okay, it tries to just find an association between how much structural variation you're seeing and variation in your trait. So here, we can't say for certain, really, even where the boundaries of this structural variant are, because there isn't one structural variant. We can say something about the genes underneath these, and there are indeed some very, very strong candidates. Almost at the end, you can also look at the effects of gene expression, and not surprisingly, you see very strong disruption of gene expression for genes predicted to be on the boundaries of our structural variant traits. So that's what's shown. Let's just focus on panel B. This is the percentage of transcripts which have no detectable expression in an RNA-seq of some of the magic lines. If a transcript spans a breakpoint, then it has basically a 50% chance of not being expressed at all. If it's inside structurally variant region, it still has about a 25% chance of not being expressed. Outside, if it's just an ordinary gene, then the probability is low. So this indicates that these, even though we can't predict the breakpoints always very accurately, that we nonetheless are getting important information out. Okay, well, the reason I came to visit Rajiv was to talk about using these sorts of approaches in the magic populations of chickpea and pigeon pea and sorghum and so forth. Also, you have a large number of accessions, so the sort of stitch type approach could be valuable there. We could think about sequencing large numbers of land races at low coverage and then imputing all the genomes. I mean, there are some issues we don't know the answers to. We don't know whether if there's strong population structure in a sample, whether that's going to affect the accuracy with which we can do all these things. Does suggest that perhaps we don't need to sequence magic founders. So if you're making a magic population and you've accidentally lost the founder, then you can still do essentially all of the work. Okay. The other attraction of this approach is that when you make a magic population and there's been some accidental introgression in the field, that will this approach will tell you that. And I think with that I will stop. Okay, just to thank people who actually did the work. So Martha did the structural variant work, Robbie wrote Stitch, the CFW mouse work was funded by the Wellcome Trust and led by Jonathan Flint, who also led the Converge work, which was also funded by the Wellcome Trust. And I'll stop there and take any questions.
Thanks very much, Richard, for very comprehensive presentation. And as you could see that he has covered different organisms and make very nice presentation that how we need to use this computational tool, how we need to dissect the traits. So although I know we have limited time, but we would like to have a few questions. So yeah, I definitely have question, but I can ask later as well. Manish, you have? Yeah.
So Richard, nice presentation, and you have mentioned about sequencing of founder parents, right?
Yeah.
So basically, if it is low coverage sequencing, or if we are genotyping with SNP-arrays, I think it will be much more effective to have founder parents, because you mentioned in one of your slide that it will be helping in the haplotypes, right?
Yes. If you're going to use a SNP array, then you should sequence the founders at high coverage. Yes, I agree. But if you, my point at the end was simply that if you don't have a SNP array and you sequence everything at low coverage, then you can get away without sequencing the founders. Although I would recommend you always do that. That's not going to cost very much. But you can then get away with sequencing the magic population at very low coverage, like less than half X is fine.
One more question. So maybe you missed to mention about groundnut magic population as well. You mentioned other crops, not peanut.
Did I miss peanut? I'm very sorry.
We already finished groundnut.
Yeah.
Okay.
So do you see that some kind of changes or modifications in the scripts for polyploid crops, or you see for polyploids?
If it's an allopolyploid, then I guess what it means is, yes, for Stitch, you're going to need to change things because you essentially have got, rather than just dosages of 0, 1, or 2, you've got 0, 1, 2, 3, or 4. So I don't know how well Stitch will impute under those circumstances. We would certainly have to change the, or rather modify the algorithm so that it realizes it's got a tetraploid. Okay. If it's something like wheat, which is hexaploid, but really it's really a diploid with three different genomes sort of coexisting, then I think it would work unmodified, he says rashly. Yeah.
So have you already used the stitch for the wheat population or not?
No, we are thinking about how we're going to sequence the magic line. So we have a 16 parent wheat population. Our plan at the moment, which may change, is we're going to use exome arrays to pull down. So we would make barcoded libraries of each of the 600 or 700 wheat magic we have. We put them into pools. We would then do pull down to focus on the genic regions, and then we would do low coverage sequencing of the pull down. That was the plan. We've just got some data back about what the accuracy of the pull down is. So we did, and that is quite a lot of off-target sequence. So we're not sure yet if that's going to be a tenable position. But assuming it is, that would be the approach that we would adopt.
And I understand that the wheat magic population already genotyped with the SNP range.
It's genotyped with a fairly low density SNP array. So I think there are about 15,000 SNPs.
Array size was 50,000 or segregation of 44?
The array size was nearer to 30,000, but the array wasn't designed for this population.
So what kind of number, or depending on the genome size, but how much?
Well, there are 60,000 genes in the wheat genome, roughly. If everything worked, then we'd have 60,000 sites. Well, it's quite likely that within a site we'd have more than one SNP. Yeah. Okay, so we might have, if everything worked beautifully, and it almost certainly won't, we might have 200,000 SNPs. I don't know. Don't quote me on that. Okay.
And by doing this 1x coverage or exome sequencing, exome sequencing will cover all the genes only, but the low coverage sequencing.
Yeah. Yeah. So the reason for the exomes is simply because the wheat genome is so large, as I'm sure you all know. It's 17 gigs. Most of it's repetitive and therefore not very informative. So it's simply to enrich. So you could argue this is sort of like a hybrid between genotyping by sequencing and low coverage.
Good. Any other questions, Prashant?
Thanks, Robert, for that great presentation. So my question is regarding the structure variations, the translocations that were identified. So you split the genomes into the 10 kb windows, different windows. So I just wanted to know, like, due to the low coverage, so what was the missing rate? Like out of, let's say you split out the genome. So what proportion of the genome was missed out while identifying?
In any given individual?
Yeah, or? Any given individual.
I don't know the answer to that question. I can estimate it assuming a Poisson coverage. So because some of the reads will overlap, you're probably actually covering, if you sequence at about half X, you're probably covering 0.3, a third of a genome, roughly, for a given individual.
So what are the chances of missing out something really important because of the low coverage?
You mean if you miss it entirely across all individuals?
Yeah.
It's
based on your experience.
It's very low from a Poisson statistics point of view, because you've got more than 100x coverage of the genome, but there will be places in the genome which are unmappable. Okay, so that's so there are blind spots, which is to do with the reference genome being so repetitive that we can't actually place reads, or at least not reliably.
And so apart from like translocations, any other specific variants, structure variants you study?
Well, we found inversions. If I can find a paper, I can tell you that.
Maybe you can have those.
Yeah, sure.
Yeah. So that's fine answer.
Okay.
This was already lost.
All right. Yeah, I'm probably going over time. Yeah, but we did break the things down by by type somewhere. If I can find a table. Yeah.
Any other question from other than these two audience? Some other young people? Monica, you have some question? Okay, you can ask one now. Use the mic, please.
How good would we stitch for GPS data, which has really low coverage and low sequencing depth?
Sorry, is your question how does this compare with GBS?
Yeah. How would GPS be? How would stitch work in case of GPS data?
Oh, I see. If you had GBS data, I think it would work. I don't think it would work to its best advantage, but it would certainly fill in any missing genotypes. Well, let me caveat that. It would work in the context of, say, a magic population where the number of recombinants per chromosome in a given individual is rather small. You know, it's 3 or 4 or something like that. So you've got a large. So the haplotype mosaic for an individual is chunky. Yeah. So yeah, you're provided you've got enough GBS sites within a chunk, you should be able to work out the mosaic. So stitch would certainly work at that level, but what you'd lose is if you haven't sequenced the founders, then you can't impute everything else. So all it will do is impute the GBS sites.
Maybe just to follow up, I don't know whether you have this plan or not, but several times when we have biparental populations, so you got only 2 genotypes, sequence them little bit higher coverage, and then you have the segregated population of the 2 parents, GBS. So probably C is asking that if we use stitch that one and then based on these 2 things, so this will be really good.
I think it would work. Yeah.
So why don't you do this thing that try to use this stitch and then compare this thing with your existing pipeline and come up with that comparison that how good it has been. Isn't it?
Yeah, I think it'd be good to try it out.
Very good. So I know that some of you will have more questions, but in the interest of time, because we already have some ongoing meeting, so let's thank once again Richard for very nice presentation. And this stitch is available in public domain, so anyone can download or
yes, it's on GitHub.
Okay, GitHub. Yeah. So you guys have not used this. So anyway, so Richard is our collaborator, so we will keep on working with him on the different possibilities. Okay, tea coffee is available. So yeah, so you are all please invited to join us for a cup of coffee and tea. And thank you very much, Richard, once again. Very great presentation.
Yeah.
Thank you.
Thank you.